Well, good evening. I'm Charles Schultz, and this is the Randomizer for March 7th, 2022. And tonight's term is going to be something a little different. It's going to be allele. And a lot of people don't know what that is. The root word for allele is allos in Greek, and it means different. If you've ever seen an alloy, you know it's different metals put together. And if you've ever seen an allotrope, such as diamonds, different forms, carbon, soot, graphite, allotropes are different forms of the same material. Well, an allele is a different form of a gene. So we're going to talk all about genes and alleles and how we know what they are and how they work. By the way, come join us as a member. Get your Oculus, get your Vive, walk around in our galleries and explore some of the things we have in there. We're adding new content. And bring a friend to show them what this is. And let's get this form of education out to everyone. It's the best form of education there is. Kids love to play in it. It's very engaging. Become a supporter, a member, or a patron of the Universe Science Center or Schultz Laboratories. Go to our page, sign up, and help us out. It's a lot of fun, and there are a lot of benefits, including these videos. Every 9 p.m. on Mondays, we have our lessons, and tonight's is the final lesson, lesson 6 of Meta World. And we're going to cover the conclusion of how we went from sticks and fire to the beginnings of nanotech and where we expect it's going to take us. We're going to take a brief hiatus after that while we get our new lessons together. We're going to be covering robotics and other subjects very soon, and it's going to be just as much fun and just as fascinating. Every weeknight at 6 p.m. Eastern Standard Time, we have the Randomizer, of course, a different technical subject chosen completely at random every day. And, of course, we're going to get more material in here and better things happening. It just gets better every day. And I'm thankful for all of our members and followers we have so far. Thank you to everybody. So, allele. Now, you know that any plant or animal that reproduces generally does so with a male and a female contributing each half of the chromosomes. Now, what's a chromosome? A chromosome is like a file cabinet or a library, and it contains hundreds or thousands of genes. And each of those genes is an instruction that tells your cells what to make. And so, when you have an organism of any sort, all of its traits are determined by the program in there, the information telling the cells what to make. Now, how do we know that? Well, there was a monk many years ago by the name of Gregor Mendel, and Mendel had a fascination with plants, and he loved peas. And whatever the reason was, in 1857, he decided to ask himself a few questions about peas and heredity in general. He noticed that in families, certain characteristics would run. So they would have a certain color hair or a certain shape of eye or nose. Everybody has characteristics, and those characteristics tend to follow in a family. So what would cause that to happen? So he asked a few very pointed questions, and he did some experiments to find out. So in breeding peas, Mendel recognized over the 8 years of experimenting that he did, there were certain traits that certain plants had. They would be either tall or short. They would have smooth or wrinkly peas. They would have white or red flowers. They would have yellower or greener foliage. And he thought about this, and so he began by taking specific types of plants and breeding them. He hand-pollinated them, carefully collected the seeds, and made sure after a number of generations that the traits were stable. So he had one set of plants that was tall, had green foliage, had wrinkly peas, had pink flowers, as an example. And he bred those plants back with the same genes for a number of seasons until he had what he considered to be a pure line. Now, he noticed something very interesting when he would plant the peas that grew from these plants. And he would notice that out of those plants, the results that emerged would be in fourths. For instance, you would have one plant that would have a trait very clearly, and it would have the same gene from the mother and the father, or the male and the female. One plant would have two genes from different parents. Another plant would have genes from the different parents in the opposite proportion. And then one plant would have genes specifically from the other parent. Now, he noticed that certain genes would dominate. They tended to express twice as often as the ones that were recessive. And so you end up with two types of genes, dominant and recessive. So let's say both parents have the genes for brown hair, and the offspring are very likely to just have brown hair. But about 1 out of 1/4 may have a variation, depending on what's in the family's history. So he decided to do these experiments and started navigating through the mathematics of genetics. And he discovered that there were actually two genes, one from the male, one from the female, that would determine what the characteristics would be, but those genes were discrete units and they would not mix. If you had a white flower and a red flower plant and you crossbred them, you didn't get pink flower plants. It didn't water it down. You either got the white flower or the red flower. If the gene for red was dominant, out of every 4 offspring, 3 of them would have a red flower and 1 would have a white flower, the recessive gene. And he figured it out. If the red gene and the white gene come from the two parents and one set of genes has red and red, that is a homozygous gene. And if you had a red and a white from probably the father and the mother, that was a heterozygous. And if you had a white and a red from the father and the mother, that was heterozygous. All 3 of those sets would express a red flower. But if you had 2 whites, that's homozygous, having the same copy, and they were from both parents, then and only then would you get a white flower. Now, this is also related to some sorts of genetic maladies from time to time. For instance, recessive genes tend not to express as often as a dominant gene, and that's true. And sometimes, keep in mind, you have more than just 2 copies of a gene. If all the copies of the gene are identical with each other, all the alleles are the same, then you have a homozygous set of genes. If any of them are different, it's heterozygous. And the numbers work out in such a way that it's very unlikely, for instance, that albino traits will show up, albinism. But if you have all recessive genes, and if by throw of the dice, out of those 4-to-1 chances, those genes express, then you get an albino organism. And so now we understand how these traits pass along, and there's a large element of statistics involved. So Mendel's experiments ran from 1857 to 1869. Yeah, I'm sorry, 1865. And he did 8 years of experiments, and in the end, he was able to qualify exactly what these units of heredity were. And he published his work in 1865 and 1869, and in a couple of journals, and it became very well accepted because his experiments could be replicated very easily. So we understand a lot about genetics due to a monk who was interested in peas and why their heredity was different. Now, sometimes if you have a dominant and a recessive gene for a trait that isn't so good, like sickle cell anemia, you end up with what's known as, in the case of a homozygous set of genes, where both of them are dominant and positive and don't yield sickle cell, you end up with an offspring that doesn't carry the gene at all. 50% of the time, you're going to have heterozygous genes where you have the gene and don't have the gene. And in those cases, you don't have the disease, but you carry the trait, and your offspring could have the disease at some point in the future, perhaps. They could pass it on to their children. If you got 2 copies, unfortunately, of that recessive gene for sickle cell, then you are going to have that illness. So now we know how these things can be passed along, just like albinism as well. And again, a gene does not mix with another gene to make a trait somewhere in the middle. You don't see that sort of behavior. Although if you have a number of copies of the gene, it is possible for more than one to express. You'll sometimes see a plant that shows a characteristic known as a sport. Now, this is an interesting thing. Some people take plants and clone them, and what they do is this is called taking an explant, and it's very easy to do. You take a specific plant that you have, and let's say it's a pea that has pink flowers, and you take a scraping of the tissue off of that plant, somewhere along the stalk or on the inside of the stalk or on the bottom of the inside tissue of a leaf, and you place those cells in a petri dish with some materials that make the cells grow, and they grow into an amount of green clumpy tissue called callus. And callus is not differentiated. The tissue grows without turning into leaves, stems, roots. Differentiation is when the instructions to the cell tell the cells what the destiny of those cells will be, what sort of structure or tissue. And so you grow callus tissue in a petri dish or in a sample, a culture medium. Now, you divide it up into a bunch of little chunks of callus tissue, and you hit them all with, let's say, rutone or one of the hormones that causes the development of roots. Now, what this does is very interesting. The moment that the root tissue starts to differentiate and grow on its own, the rest of the callus tissue reads the instructions and begins to differentiate into a stem or into leaves. It helps determine the destiny of the rest of it. And you'll get a bunch of little plants. So that's cloning the plant. And this is an easy way to perpetuate a line that may otherwise become extinct. But here's the trick. Once in a while, and it is roughly 1 out of 200 explants, will be a sport. And what that means is that at random, you've caught some of the tissue in the midst of dividing and sharing genes or changing chromosomes, and a gene got activated at random that normally would not have been. And this sport will show a different characteristic, like red stripes in the flower. Then you never saw that before. Well, it's because of something called epigenetics. There is the genetic code, as we know, which is the instructions for making proteins and enzymes and controlling other genes, among other things. And those codes have another code that overrides them, the epigenetic code. Epigenetics is a series of instructions that turns whole clusters of genes on or off to make a certain type of tissue become what it is, such as skin tissue, epithelial tissue, or liver tissue, or heart muscle tissue. So each of these has all of the genetic information in it that the rest of your body has, but it has specific clusters of genes turns on or off to determine the cell's destiny in that tissue. So in this particular case, that epigenetic information got scrambled a little bit, and the color of the flower will come out a little different. And oddly enough, you can breed these true in many cases. So one way to get new varieties of a plant is literally to clone the plant many times, and once in a while you'll see a sport come out, one that's a little bit different, and then you can take that, grow it, duplicate it, breed it, do whatever you want, and now you have a new expressed gene. Which brings me to a very interesting point. For many years, we had so much genetic information in our cells that nobody could quite identify, and it was often called junk DNA. They felt that, well, maybe it served the purpose of serving as a target for radiation damage or chemical damage, and then it wouldn't cause any trouble because the essential genes would be effectively okay. They were missed because there was another target. But that's not actually the case. It turns out that over time, you have accumulated many different genes from all of your predecessors, your grandparents, your great-grandparents, and all your greatly removed grands so far back. Many times those genes are not expressed, but under times of stress or duress, your tissue will respond in a different way. And it will respond by turning on or activating certain genes that have been actually hidden in reserve, so to speak. When this happens, new characteristics will show up, and the organism, your next offspring, will express these differences that may be beneficial to the survival in the conditions they presently are in. What's important about this is this. Many times when we try to breed custom microbes for cleaning up an oil spill or dissolving a defoliant or doing a certain task, we find that when we expose them to rigors or tough environmental changes, they respond all of a sudden. And people go, oh yes, well, they evolved or developed a mutation. No, that's not what happened. Almost never is that what happened. It's true that mutations occur all the time, and many of them are benign. A handful are deleterious or even lethal. Sometimes a gene will come along at random that is beneficial, and it's a throw of the dice. We really don't know until it's expressed. But when stress occurs, some of those genes can become activated, and those little bacteria suddenly seem to have come up with a proper response for some horrible environmental catastrophe. And yet, the fact is, those genes were there all along, but conditions in the cell, changes in chemistry or condition, for instance, acted to flip the genetic switch, and the epigenetic code was altered slightly to enable that organism to survive in tougher times. So it was an instruction that was already in their library, but it normally is not expressed. So it isn't the act of evolution, it's actually turning on, oh, I have a note here, oh, this will fix it, turning on this other instruction and dealing with the problem. So alleles are parts of our heredity, and they determine, well, whether a gene is going to override another gene, whether it's recessive or whether it's dominant. And in many cases, the combinations will be mathematically such that you'll get all recessives. Sometimes happen, or all dominants. That sometimes happens. And 50% of the time, you're just going to get a mix of the two. So, throw the dice. Even twins can have differences, believe it or not. Well, anyway, I appreciate everybody listening, and I always enjoy taking questions. And I'm hoping that more people will join us during some of these episodes and present their questions and viewpoints and give us some thinking. What's on your mind about it? Meanwhile, do go to schultzlaboratories.com or universesciencecenter.com, become a member, sign up, help us out, grow, and enjoy what you find on the site. And at the end here, if there are no questions, and I don't see anything coming up on the text boxes, I have a quotation for you, and it says, "For the Lord your God is gracious and merciful, and will not turn his face from you if you return to him." And that's 2 Chronicles 39. And that's something really good to know. You know, it doesn't take much to be a good person. Just do the right thing. Love your neighbor. Help them out. Everybody needs a hand sometimes. Even we do. And just be respectful of people. Be kind to them. And in the end, make the world a better place. And thank you for listening tonight. I'm Charles Schultz, and I hope you've enjoyed the randomizer. Come up for another one tomorrow. Have a wonderful evening. And that's it for tonight. See you tomorrow.